classification of nematodes. Types of nematodes are classified in different orders, families.
Excuse me, ma'am. Explain the last lecture's door formation once again.
Java larvae is just non-breeding, and it is a programmed, means the dormancy is programmed. So it happens in their third stage that they undergo dormancy. So if you artificially maintain at optimum temperature or whatever the environment they need for their development, then also they do not regain. They do not regain their normal life because it is already in whatever the life changes are there, they are already planned. So this is Java larvae is formed in case of dipose. Dipose is the survival strategy under which the Java larvae is helpful in their survival. Okay. So generally, I have told you so many types of survival strategies. So during their life cycle, whatever the conditions they are finding. So according to that, they undergo different types of survival strategies. Like sometimes slow dehydration is there. Sometimes fast dehydration is there. So in both cases, different types of survival is there to regain their life structure again when they find their suitable condition again. Okay.
I mean, it's a kind of irreversible.
Yes, irreversible.
So,
Irreversible means if you provide the temperature and moisture and whatever the conditions they need, again they will not regain, means they will not come in that normal position. But when according to their program, like in Anguilla triticum, okay. So Anguilla triticum is a paste of wheat and barley. So wheat and barley is a Rabi season crop. If you plant that crop, providing that situation in off season, then also these nematodes do not hatch from their cyst, from their galls, because they have the plan that they revive them, they hatch from their eggs when the season comes. Okay, so during November, December, they will again revive. Got it now?
so I mean, in this case, the pattern of life cycle is completely changed.
Yes. So because nematode is also considered as an animal because they have all the development structures and they have well adapted to the environment. They have very well developed ecological behavior. So that's why they are very much important. And you know that only 10% of all the nematodes are plant parasites. So what about the 90%? These 90% nematodes which are abundant in our environment, and they are very like saprophagous nematodes, all the marine nematodes, you know, this is two-third part of our earth is filled of water. So that water contains this marine water or fresh water. So these nematodes are very helpful in recycling of waste materials, then pollution indicators, then mineralization of what this soil microbe, then decomposition of different residues. So they are very helpful in our environment and for our some nematodes you are not going to study in this course, but some nematodes are also beneficial, like they are Uh, they are used as killing of our crop pest, is a very important crop pest, like entomopathogenic nematodes, like grasshoppers, then mosquitoes. So these nematodes are found everywhere because of their adaptation, because of their survival, and because of their well-organized body. So in this way, nematodes are very interesting organisms. So have you been to YouTube on C. elegans or biological studies? Anyone have seen? Last time I have told you to go through on, because this C. elegans is a model organism, and many type of biological studies have been done on this organism. Other organisms are Panagrillus retivivus, and some other nematodes are also there. But C. elegans is very important, and because of their small size, because of their short life cycle, and high fecundity, easily cultured in the lab, easily stored at deep freezer. So whenever you want, you can take that organism and you can study. So now today's class is, can you see the slide? Hello, Abhishek.
Yes, ma'am.
Okay. So in classification, you know that nematodes are of different types, and their diversity is so much that they can parasite anyone. They can parasite plants, animals, human beings, fruits, fruit trees, and small insects. So the need of, you have studied in biological sciences. So in biological science, whatever the organism is, we have to classify according to their morphological structures. So on the basis of that, nematodes are also classified, and some important terms before that we have to study, like taxonomy. Taxonomy means taxa is the different stages of that classification. So we'll study later on. So taxonomy is the science of classification, including its basis, principles, procedures, and rules. So whatever means organisms are there, so you have to classify them. And on what basis you are classifying, on what principles, what the procedures you apply, and what are the rules. Because if you don't follow that rules, you cannot classify but certain organisms. Like you find some organisms, and on the basis of their characters, you have identified that these have this type of tail, this type of stylet, this type of esophagus. Then how do you classify in certain taxa? So there are some rules. So according to that rule, you have to classify them, and you have to report them to the taxonomical bodies. So otherwise, you cannot place them on that taxa. So it includes the identification of specimens, publication of comparative and descriptive data. Publication of comparative and descriptive data means there are already identified nematodes that are classified under different groups. And suppose you have identified some nematodes, then you have to compare with the other nematodes which are more or less same or similar to that organism you have identified. So that you have to produce, that you have to publish that data in descriptive way. And the study of diversity and relationships among organisms. If there is some difference you can identify, then you have to report them, you have to publish them. Then again, it will be included in that organism, in that taxa. Okay, so another term is systematics. Systematics is the broad term. Systematics is the broad term, which is also a scientific study of principles of classification of organisms using all available biological data. Whatever the
The slide is
biological,
not changing.
Now,
No, ma'am.
oh, I'll have to again share it. Okay, just wait. Now came
No, ma'am.
systematics.
No, ma'am. Yes, ma'am.
Now came. Is it okay?
Yes, ma'am.
So, systematics is the broad scientific study of principles of classifications of organisms using all available biological data. It deals with kinds, diversity, and the evolution of organisms into groups and their systems. So with their kinds, diversities, relationship that we have identified with the comparative data and the evolution of organisms, how they evolve. Actually, nematodes, I told you in last class that they have evolved through different types of evolutionary, like different types of environmental factors in which they are surviving. So suppose the nematodes are previously only the saprophagus nematodes, very large, very thin, and they don't have a style, they have only parts. So they just swallow the microorganisms and survive. So after that, when sometimes they don't find food, they they get across with some animals, some insects, some plants. Then during breeding, they adapted themselves to that organisms, and that organism or that plant become their host. So they get adapted with that. So this is the evolution. So according to evolution, they are classified and their arrangement to groups and their systems. So systematics actually deals with the diversity and the study of population, species, and higher temperature. It plays an important role in both applied and theoretical biology, and it is the only science which deals with the development of a complete understanding of nature and its diversity. So that was because taxonomy and systematics, these are very much important, because if you don't place in a certain group, um, certain taxa, then you have to identify them according to that group. So what are the biological behaviors, what is the physical behavior, what is their feeding behavior. So according to that group you can assess. Now the classification, is it visible?
Yes, ma'am.
So classification is the procedure, means you have to classify different organisms into different groups. So this is classification, and classification is again on the basis of different characters, which is two types, like artificial and natural. So classification is the ordering of organisms. You just place them in different groups, into groups and systems based on their relationships. Such relationships can be either phonetic, phyletic, or both. So phonetic means their morphological characters. Phyletic means their relations with the other already identified organisms. Means you are having father, grandfather. Then some morphological characters are there that somebody who are not knowing you, then they can identify on the basis of your file morphological character that you may be the grandson of that person, you may be the son of that person. So that is the phonetic. And phyletic means according to your behavior, according to your
Excuse me, ma'am.
attitudes. Hello.
Some students wants to join.
So that is the disturbance. That's why I wanted everybody should join for the class.
Yes, ma'am.
So on the basis of these characters, generally people grouped into different groups. So there are two types of classification, like artificial and natural. So artificial basis, we have to the organisms may be arranged in any manner necessary to achieve a desired directive. For example, pictorial key used for teaching and identification. So these artificial classification changes every time whenever you need. So according to need, you can group them in different groups. Like some nematodes are endoparasites. So in endoparasites also, like that Melidogyne incognita are sedentary endoparasites, but some endoparasites are also migratory. So according to their behavior, you are grouping into that group. And sometimes you find that these migratory nematodes, they are in different families, they have different characters. And these endoparasites, sedentary endoparasites like cyst and root gall nematodes, they have different characters. So they are grouped into different um, taxa. And in natural means already they are having in that they are having characters which are found in other group. So it includes the natural characters of organisms. So that is the natural classification. Taxon. Slide is visible. Jyoti.
Ma'am, classification अभी visible है. Yes, ma'am. Now it is visible.
So I will keep in that so that you can see. Taxon. Taxon is a taxonomic group. So you have might have heard about that different taxa is there, like phylum, families, subfamilies, orders, class. So these are called taxa. So these taxa may have higher taxa or lower taxa. So from the species level, they are lower taxa, and above the species level, higher taxa. So this is taxon, actually a singular word, and taxa is a plural word. So taxon, what is taxon? A taxon is a taxonomy group of any rank designated as a definite category, like species, genera, families, orders, classes, and phyla. These are all examples of taxa. And now the species. So from the species, you may find subspecies, then varieties, then pathotypes, biotypes. I told you in ecology class that some are behaving, some individuals of particular, some behave differently in their biological behavior. So they now again they are classified into biotypes, pathotypes. Some are classified into races. So these all taxa are called lower taxa. So this is actually species is the most important taxa in biology because in general you can find many types of different behavior. So according to that behavior, species are classified. So these species may be genetic species or phenotypic species. Genetic means on the basis of their genetic character, and phenotypic means on the basis of their morphological or phonetic characters. So genetic species, it is a taxon composed of groups of interbreeding natural populations. So if the genetic species are there, they interbreed in their natural population which have similar genotypes but are reproductively isolated from other such groups of natural population. Means if in their population some individuals have different genotypes, then they don't interbreed, but they interbreed between the populations having similar genotypes. Then phenotype species, it is a taxon composed of groups of natural population having a high degree of similarity in their phenotypes. Means morphologically they look very much similar to each other. Then the phonetic or phenotypic species. So you already know that these are morphologically very close. So taxa described at the specific level from any type of biological data other than that pertaining to the genotypes and reproductive isolation. So most of the described species fall in these categories. Generally, whatever you classify, they are classified on the basis of their morphological characters, like what type of cuticle is there, what type, how many lateral lines are there, how many um, papillae are there, how whatever what are in the characteristic of plasmids, amphids. So these are all the morphological features on the basis of which you identify. So generally, phonetic or phenotypic species are considered for their grouping. The genetic species are not very useful in taxonomy. So on the basis of genotypes, generally we don't group them because they have, again you have to find their genotypes. So that is a cumbersome job. So phonetic species dominate the nematode taxonomy. So on the basis of morphological characters, we generally study their behavior and their taxonomy is grouped. Now the species on the basis of diversity. How do we classify the species on the basis of diversity? Like monotypic species. If population of a species have very little variability, if suppose in any population like Melatogen incognita is there, then some nematodes they may have different morphological, very small morphological differences. So then they are called monotypic species. If polytypic species are there, if the group of populations have a great degree of phonetic variations, for example, biotypes, races, or subspecies. So these subspecies, these races, suppose Melatogen incognita is there, then they have different races like race 1, race 2, race 3, race 4. So on what basis they are classified? So I have given you that table. You can go through it again. So that races are divided into different races because of they reproduce on different plants. Same nematode, same species. So according to that, they are classified into different races. So they are called polytypic species. Sympatric and allopatric. So sympatric and allopatric species, like when the geographical changes of at least two species partially or entirely overlap and the population do not interbreed, the species are sympatric. Like in certain areas, Melatogen incognita is found. Okay, and after that, uh, in certain areas, Melatogen javanica is found. So these both are two different species, but in the adjoining point there are some overlapping. Means in one part there may be javanica and incognita both are found. In the adjoining areas of other part both are found. So this is somewhat mixing of that population at certain point, but they do not interbreed because javanica do not interbreed with the incognita. So these species are called sympatric species. So at that point, both the nematodes are found, like Melatogen incognita and Melatogen hapla in Canadian greenhouse. So in Canadian greenhouse, both the nematodes are found, but they don't interbreed. They have to find their own species for their further population. In Georgia also, in their cotton soil, Trichodorus cresti and Trichodorus porosus These two nematodes are found having the same genera but different species, and they don't interbreed, but they are found in the same soil, so they are called sympatric species. Allopatric species occur in non-overlapping but often adjacent geographical zones, like Globodera rostokiensis and Globodera pallida. You can write here Globodera rostokiensis and Globodera pallida, Pallida. So Globodera rostokiensis and Globodera pallida, they both are of same genus, but have different um, species. So they are, if they are found in adjacent areas, then they are called allopatric, means they are not mixed, but they are found in different geographical areas very near to them. Then how these species evolve? Okay, so any new species, if you find that before describing any new species, you have to go through that, how they evolve. So species undergo four stages during their existence, like new species, major species, new species, and tilo species. So neo means very nascent, very new. This is the first stage of evolution when they increase their abundance. Suppose some populations they are find some host, some plants that their roots are very much in abundance, so they evolve there. Means they multiply in abundance and they evolve some characteristic, and they expand their geographical distribution. Means if the population is high, they spread their population to the surrounding areas. So this is the neo species. And major species, so this is the second stage. After spreading to the new geographical areas, they do whatever they do extensive interspecies variation. Then there, among their same population, some variation will be evolved. Means whatever the original will be for the first stage of evolution. So whatever the characteristic of their original species, they have developed some different characteristics. Okay. So relatively great abundance, again they are in great abundance and they have geographical boundaries. Now their geographical boundaries are there, means after their spread, they capture some boundaries, they dominate some geographical boundaries. So now they are called major species. After that, the third stage is new species where the if you are finding that this organism was not really attacking on that plant, now these are very much common and they are found in a particular areas. Like any particular crop, particular area. So if previously they are not found, but now they are very common. Whenever you go, you can find them. Then it is the true species. So they have fixed geographical boundaries and intraspecific variations are minimum. Means among those the intraspecific variations are minimum. So that when the intraspecific variations was high, that time it was separating from the original population. And in new species, they all are new and they within them the intraspecific variations are very less. Then the tilo species, when they reach their final stage, that is narrow geographical distribution, not very abundant. Now they are not very abundant and they are a very narrow geographical distribution. Now they become tilo species. So this is the evolutionary stages of any species development. Now the higher taxonomy characteristic, I told you that from up to genera, phylum to genera, all the taxa are higher taxa. And from the species level to the all the lower levels, they are called lower taxa. So higher taxonomic category is a taxon above the specific level, species level. Linus, you might have heard about him. In 1758, he was the first to establish a definite hierarchic taxa of animal kingdom into 5 taxa. That is classes, orders, genus, species, and varieties. So species and varieties, they are lower taxa and these are higher taxa. So phylum is the highest taxon. So after phylum, class is there. After class, subclass, then order, then suborder, then superfamily, family, subfamily, and after subfamily, genus. So there are some suffix. So if you have to know that what type of taxa is there. So according to that suffix, you can find that this is order or this is suborder, whatever is that. So for order, it is da, like Tidenkida, Aplenkida, Torilamida, uh, Diplogasterida. These are orders. And order, in order, different suborders are classified. So they are suffixed with na, like Tidenkina, Aplenkina, Anguina. Sorry, not Anguina. Tidenkina, Aplenkina, and others are there. Superfamily Oidea, like Tylencoidea, Hoplolemoidea. So they are the lower taxa of suborder when they are again grouped into and differentiated into different families. And family is GAE. It is GAE, like Tylenkidae, Hoplolemidae. It means it is a family. And under family, if the subfamilies are there, then they will be suffixed with ni, Tylenkini, Hoplolemini. Okay. Now this is a classification which starts with the phylum. So this is again a very large uh, line diagram. You can see, call it a flow chart. So you can see that up to subclasses I have written here. So phylum, in phylum Nematoda, two classes are there, Phasmidia and Phasmidia, or you may call it Adenophoria and Ciccianentia. So during that morphology class, I have told you about Phasmidia and Phasmidia, that the nematodes are grouped into two groups. One group is having phasmids and one group is having absence of phasmids. They are not having phasmids. So that the primitive type of nematodes, they don't have phasmids. Means they don't have that paired sensory organs that are found on the tail, and it is also called adenophoria. So adenos, ADENOS, adenos means a gland. So they have glandular type of excretory system. So on the basis of that excretory system also you can group into this into two groups like adenophoria and Cicerneria. Cicernier, Secerneere, Cicernier means tubular like. So if the excretory system is tubular type, then it is grouped into Cicerneria. Again, these classes are classified into subclasses, like Phasmidia is classified into Enoplea and Chromadoria. Okay, these two subclasses are under Phasmidia. And in Phasmidia, three subclasses are there: Bacteria, Diplogasteria, and Spiroria. So amongst all these, this Diplogasteria, it is very important, and it is having all the plant parasitic nematodes. Means most of the plant parasitic nematodes are in Diplogasteria. Rhabdicea, they are all of saprophagous nematode. They are found in water. So whatever it will be, marine water or fish water or in soil also, they are found. They are very common. Spiroria, they are all insect parasites or human or animal parasites. In Enoplea and Chromadelia, we will again find some growths which are having plant parasites. Some are having plant parasites, some are predatory. So we'll start with one by one. So this is orders of Inoplea. Inoplea, you just remember that in Inoplea, two orders are there, Dorylamida and Triplonchida. I told you that this taxa will have TA suffix, Dorylamida and Triplonchida. So this abbreviation is FW means freshwater, MW means marine water, and BW means brackish water. Brackish water, when the river left to other direction, then the leftover water is called brackish water. So this Inoplea is having this 1, 2, 3, 4, 5, 6, 7, 8, 8 orders, but we have only Dorylamida and Triplonchida, which are having parasitic nematodes, PP and plant parasitic nematodes. So we will consider them 2, 1. Now the others are Inoplida, Isolamida. They are having soil habitats, bacteriophages, some are predators. Mononchida, I told you these mononchida are very important because they most of these nematodes are predators, but some are free living and soil nematodes also. Myrmidida, I told you about myrmidids. They are very thin and can be visible through your naked eyes. So they are insect parasites. They can feed on grasshoppers. They can feed on mosquitoes. Myrmis nigricans, you have heard about the name. Then these are Myrmidida. Then Trichocephalida, they are vertebrate parasites. Musca pigicida, these are vertebrate parasites. So vertebrate parasites means they are feeding on animal or human beings. The second one is Chromidoria. Chromidoria doesn't have any plant parasitic features. So only this Inoplea is important in which case Adenophobia. Means in primitive nematodes only the Inoplea is important in which you have to study about Dorylamida and Triplonchida. So this Chromidoria is having 5 orders: Chromidoria, Adenomida, Monistorida, Desmodorida, Descosorida. So you don't have to remember all this, but just you have to know that in Inoplea, Dorylamida and Triplonchida, which are plant parasitic. Now the third order is Rhabdicia. Third class is Rhabdicia, which are you find here Rhabdicia under Plasmodium. Okay, so Rhabdicia is Rhabdicia also do not have any plant parasites. They have three orders: Rhabditida, Ascarida, and Strongyloida. Ascarida is the compound that it causes diseases in human parasites. So these are parasites. Strongyloida, they are also parasites. Rhabdicia. In Rhabdicia, there may be soil parasites or bacteriomonas or vertebrate or invertebrate parasites. So these Rhabdicia are very well developed, but they don't contain plant parasites. Now the Diplogasteria. This is very much important. I told you that Diplogasteria is one of the most important plant parasitic. So in Phasmidia, that is very advanced group, is having Diplogasteria. So we study Diplogasteria. In Diplogasteria, 3 orders are there: Diplogasterida, Dilenchida, and Aplanchida. So again, these 2 orders, only these 2 orders are plant parasitic nematodes. So we will study only those 2. Diplogasterida, they may be bacteriophages, they may be predator, they may be vertebrate or invertebrate parasites. Now the next one is Spiroria. Spiroria means again in the case of here Spiroria in case of Sissionia. So in Spiroria, 2 orders are there: Spirorida and Camelida. So Camelida are all these parasites, and Spirorida are vertebrate or invertebrate. So we don't bother about that. We go to only the plant parasitic nematodes order, that four orders like Dorylamida, Triplonchida, and then Palanchida and Aplanchida. So now the characters of Nematoda. What are the main characters, major characters of Nematoda? That is phylum, and this is the highest character. So previously nematodes are grouped into different elements and collecting elements. Now they have classified into different group. That is phylum and nematode. So nematoda animals are bilaterally symmetrical animals. Bilaterally symmetrical means they can be societal plane which they are two identical uh, parts of their body, and they have radial symmetry in the region of head and pharynx. They are generally small in size, worm-like and circular in cross-section. Circular in cross-section means when they cut the CS, will get circle. They are aquatic, terrestrial, and free or parasites of plants or animals. Their body is unsegmented, unlike the earthworms. So, if that segmentation is present, it means there are cuticle ornamentation. So, because of that, it looks like segmented body, but it is not segmented. Elongated, they are very long, cylindrical, vermiform means they are tapping, their body is tapping towards both the ends, mouth end and tail end, and more tapping towards the tail end. Cuticle of the body may be smooth or involuntary. Respiratory and circular system absent. You already have seen that. Male and female are found separately. That is diagnosis condition means they both have different individual characters and they are different. Since there are some exceptions where male and female characters are present in one organism that are rare. Sexual dimorphism is absent. Sexual dimorphism means both male and female are different morphological features. Like in Melatog and incognita and cyst nematodes, females are globose shape, females are round shape, and males or juveniles, they are spindle shape. Vermiform. Now the first class is Adenophoria and Sissionintia. So in Adenophoria and Sissionintia, you have grouped into according to their plasmids presence or absence or their expiratory system type. So in Adenophoria and Sissionintia, I have written only 10 characters here. Otherwise, more than 10 characters are also there, but these are very important characters which make them in the different groups. Like plasmids are absent here, the plasmids are present. So you can remember here also that A for Adenaria and A for Plasmidium. So both start with letter A and they are both similar. So adenos means gland, a glandular shaped excretory system, and Sissionintia means they have a tubular type of excretory system. So here plasmids are absent, here plasmids are present. Caudal glands are present, here caudal glands are absent. So there is actually whatever the organs are present, if they are not in much use, they are finished, and in advanced organism they are not found. So these caudal glands are not much very important, so they are absent in the Sissionintia class. Diorchic testis. Diorchic means two testes are present. Here only monoorchic testis. So that another testis is the called posterior. If it is present on labia, then it is called labia. So in Sissionintia, it is present on the legs, and in Adenophoria, it is present behind the legs. Derids absent. Derids are also some type of sensory organs. These are also present in pair, but they are absent in adenophoria and present in Chichen-Itchy. Bursa is absent. Here bursa is present. Ventromedian supplements present. Ventromedian supplements means they are present some type of sensory organ that are present on the tail region. So here it is present and here it is absent. Cephalic papilla 4 of 16 is city-like. Cephalic papilla told you about the morphological features of these sensory organs. So cephalic organs means they are present in the cephalic region, that is esophageal region. So they are CT-like and they are 4 or 16. And here cephalic papillae are not CT-like. Not CT-like. Here cephalic papillae is not CT. So generally what happens when the CT, this papillae is just like a pimple type, and when it becomes CT means uh, there is a stick type structure on that papillae. So it helps in their movement. It helps in the movement in the water. So most of the adenophories they live in water. So they need swimming. So they have cephalic papillae. Opening of subventral glands is near the nuclei. Opening of subventral glands is away the nuclei. What does it mean? Opening means you know that in esophagus, that basal wall is having some esophageal glands. One is dorsal esophageal gland, and two are subventral glands. So, in case of adenoria, there are two pairs of subventral glands. Means four subventral glands are there. In Cicinacea, only two subventral glands are there. So, what happens in case of during feeding? Only first the dorsal esophageal gland secretion is there, then the subventral. So here the subventral glands open near the nuclei. Nuclei means that basal part, the basal part. So that subventral glands just open in that basal part, and here it is away from the nuclei. It is not opened in the nuclei, but it goes to the upper part. So after digestion, partial digestion of food, when the food comes inside, then subventral glands secrete their enzymes and it is away from the nuclei. The inner lining at the opening of this petri pore is not cuticularized. So cuticularized means that skin, that cuticle is having smooth or they just protect the skin of nematode. So whenever there is a pore, like expiratory pore, vulva is there, mouth is there, anus is there. So on this particular type of pores, this cuticle goes inside to line it with the cuticle so that during expiration, yes, it should be no problem. But here the inner line with the opening of the pore is cuticularized. Here it is not cuticularized, here it is cuticularized. Now the first order is Dorilamida. So we'll start with Dorilamida. So Dorilamida, you know, they are very large nematodes. You can easily find that they are having which type of esophagus is having Dorilamida. Any one of you remember that
Ma'am,
esophagus?
two-part
Two part.
esophageal
Two
system.
part. Very good, very good. What's your name?
Ma'am, our frame.
Amar. Okay. So Dorilamida is having two part of esophagus, but whatever the line diagram you have seen, that you may not see in live organism because they are organisms, their parts may be distorted. So in some nematodes like Giphinema, Dorilemus, and then Longidorus, they are very large nematodes, very long nematodes. So their esophagus is also very long. And there, esophagus, you told that two part is there. So one part is procarpus and posterior part is postcarpus. So that postcarpus is also very long, but that procarpus, they are thin and very long, sometimes in folding condition also. Okay, so that way you have to, when you see the dory laments, you have to identify uh, what type of their esophagus. So on the basis of that, you can identify that these are dory laments. And one more characteristic is that for dory laments, that they are having this type of stylet is there,
Ma'am,
odontostyle,
odontostyle.
odontostyle. So this is the characteristic. These two are very important characteristics. Like they are having two part esophagus and odontostylitis. So odontostylet and stomatostylet that are found in Thailand. So what are the difference? Why they are called odontostylet? Why they are called stomatostylet? So what happens that in odontostylet that the dory lamina is having, they are formed at different places from their stylet. There is a gland through which the stylet is formed. Already in formation and one is that is in function. So that is called functional odontostylet and that is called replacement stylet. And during molting, again the third stylet is there, that is molting stylet. So in this way you can identify that this is molting nematode, means these are molting their cuticles. So the molting stylet is there. One is replacement stylet and one is functional stylet. And if the nematode is having only one stylet, it means they are adult. Means after getting adult, they don't need to molting. They don't need another stylet. So they are having only one stylet. So this is the identification. And these nematodes are having very clear cut stylet recognition. When you see under microscope, you can easily see that two stylets are there or three stylets or one stylet. So these dory laments, they are very common in soil. Whenever you wash any soil, you can find them. They may be either predator or bacteriophagous, and some very few are plant parasitic nematodes. So we will study only the plant parasitic nematodes. They are only ectoparasite. They don't go inside the root. Their size ranges from 0.3 mm to 13 mm. So 13 mm, you can imagine that one small thread-like structure. If you uh, carefully see, you can see without microscope that these are thread organisms, small thread-like. But you cannot see all the parts of that body, that you have to see through microscope. Both the sexes are vermiform, means here males and females both look alike, both are similar in morphology. No sexual dimorphism except at the tail region of male reproductive system. So I told you that male nematodes, they are having secondary sexual characters at their tail, like spicules, gubernaculum, and versa. So because of that, it differs from the female. Otherwise, there is no differentiation. Other characters are that amphidial apertures are pore or slave-like. Amphidial structures, I told you that amphids open through different types of structure, like sometimes it is a pore, sometimes it is a slit, sometimes it is spiral shape, sometimes it is a horseshoe shape. There are many types of opening structures. So here it may be pore or slave-like. Amphid shape is also variable. Stoma is provided either with neural tooth, odontostyle, or they may be astomatous. In some nematodes, there is no stoma. In some nematode there is neural tooth. Neural tooth is a solid tooth, and some nematodes they have odontostyle, and they have 5 esophageal glands. I told you already that the primitive nematodes, they have 5 esophageal glands, one dorsal esophageal gland, that is DOG, and 2 pairs of subventral glands. These glands open in the bulb. Bulb is that basal bulb, that the third part of, sorry, here is the second part of esophagus. Females are either amphidelphic. Amphidelphic means having 2 ovaries. Monoamphidelphic means 2 ovaries and both are directed anteriorly and posteriorly. Monoprodelphic means 1 ovary that is directed anteriorly. And monoopisthodelphic means if one is present and that is present near the tail region, then it is monoopisthodelphic. So you can see here in this diagram, this is the this is the esophagus. So one part is here after stylet. This is the stylet region. This is the procarpus. This is the postcarpus, also called basal bulb. So in basal bulb, five glands are there. Here it is. The first one is larger, which is dorsal esophageal gland, and in posterior region, 2 pairs of subventral glands. So this is subventral esophageal gland. This is subventral esophageal gland, and this is the last one, last pair, subventral esophageal glands. So you can see here, dorsal gland opening is here, and these two also open here near the lumen. This is the lumen. It is a hollow structure through which the food passes through. The ovary is always reflexed because you know this is a large nematode having to produce many eggs, so their ovary size is also very long, and because of long length, they reflex. Reflex means they are in folding state; they are not straight. Okay. Female tail is quite variable. Male diaortic means two testes are there, one directed anteriorly, other posteriorly, and males are without bursa. Males do not have bursa. Genital papillae. Genital papillae means on the tail they have some papillae. They are sensory structure just to find out the mates, and they are intromedial position and anterior to the anus, means they are found just above the anus. So you can see here in this diagram that they have reflexed ovary. Means this is the ovary, but they are folding. They are folding here, and this is the germinal through which the egg multiplication is there. This is diode. This is the testis. One testis here, one testis here, and from here the sperms will come out. So this is called diode testis. Now you can see here the tail part of male organ. Through lateral view, means from side you can see that one spicule you can find, and below the spicule you can find a gubernaculum, which is a very hard plate-like structure. So this is called lateral guiding piece, which is gubernaculum, support spicule from lateral side, and this is the ventromedial supplement. This is the papillae on the present of the tail is to find the mate. If you see this tail, the male tail through ventral side, if you see on the ventral side, then you can find the ends. You know, all the openings of nematodes they found on the ventral side. So if you see through ventral side, these nematodes still having anus, you can find it easily, and have two spicules. One is here, one is here, and here the supplement that is a small one. You can find. Now the Dorilamida order is grouped into 3 suborders. So suborder is having NA, that is Nicolamina, Dorilamina, and Algamina. So Nicolamina, actually these Nicolamina are having all the predatory nematode. They are having neural tooth. So I told you that in this group either they are having neural tooth or odontostele or ostomatous, means they do not have stoma. So that ostomatous is grouped into Albumin suborder. Those are having odontostylates, they are grouped into Dorylamina. So these groups are having all these plant-parasitic nematodes which are having odontostyles. So through odontostyles, they puncture the plant cells and feed on them. Nigolamina, they are having neural tooth, so they can predate on other nematodes or other microorganisms. And their cardiac glands are free. This is a peculiar character. So you can see these are the cardiac glands. Cardia, after esophagus, intestine starts. So between esophagus and intestine, there is a wall. So they can open at one side only, and this is called cardia. And below the cardia, three glands are there. So these are called cardiac glands, which are present only in nigolamina. That is the predatory nematodes. And this is neural tooth, just like a long tooth. This is found in algal. No, sorry, nigolamina. Now the order, suborder Dorylamina. They are having plant parasites. So they are again grouped into Dorylaemoidea, Belonodoidea, Dylencoraemoidea, Actinolemoidea, and Longidoroidea. So you don't need that much in detail, but just to know that Dorylamina is having plant parasitic nematodes and they are having Dorylaemoidea, Belonodoidea, Dylencoraemoidea, Actinolemoidea, and Longidoroidea. So here only Longidoroidea, they are having plant parasitic nematodes. In Longidoroidea, only three esophageal glands, posterior pair is missing, but opening is present. Only this superfamily contains plant parasitic nematodes. So again, you have to remember only Longidoroidea. So in Dorylamina suborder, only this is Longidoroidea. So you just see that OIGEP. This is the suffix of superfamily. So this is Longidoroidea is the superfamily. So these are the different types of, you need not to know that all in detail. So in Longidoroidea. So Longidoroidea you have to know they are large size nematodes like 1.2 to 30 mm. Both sexes vermiform. Sexual dimorphism is missing at the tail. Cuticle is without any annuals. Annuals means cuticle is without any annuals. Annuals means that cuticle has some folding like structures that is transverse striations. So because of that they look annulated. Ampid is pore-like or slit-like. They have long stylet. You can see here 40 to 200 micrometer, known as odontostyle. At the base, the stylet may be bifurcated or forked. Guiding ring may be bifurcated or single. Stylet extension may be with swellings or not. Stylet extension, you know that stylet is divided into three parts, that is conus, shaft, and stylet knobs. But in case of odontostyle, odontophore and odontostyle is there. Odontophore is that is upper part of that stylet and odontostyle is below part. So they may be with swellings or not. So if the swelling is there, there is different nematode. If the swelling is there or not there, they are different nematode. Here you can see this is the ampid. These all are ampids. These are having slit type of opening. These are having four type of opening, and these are having large slit type opening. This is bilobe. So here you can see the stylet part of Dorylamida. You can see this is very long. The upper part is odontostyle and lower part is odontophore. And this is the guiding ring. Guiding ring is present. Females may be monoopisthodylpic or pseudomonodylpic. One mobile is not complete. Female tail is quite variable, long and filiform. Males are diarotic with medians appearance. And now they are having three families. So up to Dorilamida, I will finish today. So they are having again classified into Longidoroidea, that is superfamily. The superfamily is classified into three families. The family name ends with DAE. So remember that. Jiffy Nematode, then under this family, Jiffynema species. This is the genera Jiffynema, genus Jiffynema. Under Longidoridae, Longidorus and Paralongidorus. So in Longidoridae, two genera is there, Longidorus and Paralongidorus. And they may have many species. So if you write many species, then you have to write SPP, and it should not be in italic. If you write the name of species, then it should be in italic. Then the third family is Geoffidoridae. Geoffidoridae is having the genus Geoffidorus. Okay, so Geoffynema, Longidorus, Paralongidorus, and Geoffidorus. These four nematodes come under the group Longidoroideae. So what is the difference between Jiffy Nematode and Longidoroideae? So if some nematodes are grouped into this one and this one, it may be because they have different characters. So what are the different characters? So this having amphibial aperture is snake-like, here it is pore-like. Pore-like, but maybe bilobed or bilobed, single-lobed or bilobed. Here the base of odontostyle is bifurcated, here it is not bifurcated. Odontophore is having flanges. So here the flanges are present, here flanges are not present. Guiding ring is double and is present near the junction of odontostyle and odontophore. So I have seen now that in diagram guiding ring is present. So here guiding ring is double, here guiding ring is single, and at lip is. Now what about the basal bulb? Basal bulb, the dorsal gland is larger than subentrance and its opening is very near to the lumen. But here in case of Longidoroideae, dorsal gland is smaller than subentrance and is located away from the subentrance. So here all the five glands are very near to each other. Here the dorsal esophageal gland is far from the subentrance. Here in Jiffy Nematode, females are amphidelphic and pseudomonodylphic. Here the females are always amphidelphic. You can see here that this is Jiffy Nematode and this is Longidoroideae. So here in case of this dorsal esophageal gland, this is larger than these two, and it opens just in the lumen, just near the lumen, and it is having a small duct through which the secretion comes here. But here you can see that this opening is not very clear near to this subentrance. It is away from the subentrance. So this is the difference between these two. Now the Geoffidoridae. Geoffidoridae is having the characters between Jiffy Nematode and Longidoroideae. So amphids are of Longidoroid type, odontostyle of Jiffy Nematode type. So here the difference is that guiding ring is single and it is away from the that part. Basal bulb is Longidoroid. So again, you can see here that in Longidoroideae, two genus are there, Longidorus and Paralongidorus. So why they are called different with different name? Because they are placed in same family. So may be having there one or two differences. So what are the differences? Longidorus and Paralongidorus. So here the amphids are four-like, here the amphids are shape-like. Here the ampid shape is lobed or bilobed, here it is stirrup shape. So these two differences are there between Longidorus and Paralongidorus. Now the very peculiar character of Dory So why they are grouped into plant parasitic nematodes? Actually, they are responsible for transferring of virus from they act as vector. They just carry the virus from one plant to the other plant. So the juveniles, that first, second, third, and fourth, before becoming adult, they have two stylets that I've told you. One FO, that is functional intestine, and the other RO, that is replacement intestine. Adult has only one stylet, that is functional intestine, and the molting stages have three stylets, that is FO, RO, and MO. MO means molting style. So these all have different types of stylets in their body during their different life cycle structure. Now, because of their different structures, they carry the virus in their esophagus at different part. Like here, you can say this is J1. Okay, so this is showing the stylet structure. Okay, this is J1 having only one stylet. This is functional horizontal state. If it is J2, J3, J4, then the other stylet is manufacturing here with the gland and the functional horizontal state. So when this functional stylet is small, then this stylet will come here. Then again the synthesis is completed. So you can see here the tape-like structure that is manufacturing here. You can see okay, so next time we go on triple beta, that is really no order. Then we use the video for the session. So these are all Indian languages. I hope you have enjoyed. I hope the classification is more to understand the deliver. So if you go through the slide frequently, so okay, anyone having any questions?
Excuse me, ma'am.
Yes.
Ma'am, if birds are present in well dormant limits, then which effect there is help in holding the female during population?
They are actually married or silent or sometimes they are very large animals, so they don't need the actual population. So they are different. Okay. We have seen that control subsidies. So they say they are the population. Okay.
Okay.
So I'm closing this.